are motivated by an interest in how the brain is processing information. All of us are interested in what addiction means and how an addicted brain functions differently. I think we're all interested in understanding something about the way in which animals are processing information and the way the nervous system functions as a biological computation decision-making entity. The next half of this story is really totally a different focus, which is to say, not that we're done with the behavioral pharmacology, but if we know what we know, can we exploit that for therapeutic gain? Can we learn something? I told you about all of the bad things dynorphin does as a stress molecule. Will kappa receptor antagonists have therapeutic potential? And so I'm excited about that idea because I think that animal models of human behavior are always limited, and it'll be much more exciting to be able to do some of these studies with human models. And to do that, we've got to get drugs into people. And the kappa antagonists that have been developed recently seem very encouraging. As I told you earlier, when the different types of opiate receptors were described, mu, delta, and kappa, based on radioligand binding studies in the early 80s, the drug companies got all excited and they developed all of these selective kappa agonists, but none of them had any compliance. None of them were very good because they all had compliance issues. And so that really kind of put a halt to anything more in terms of drug development. But when we found that norBNI could block stress-induced potentiation of cocaine reward, it rekindled interest in the idea that kappa antagonists might be able to promote stress resilience. And I described to you how we were able to separate the P38-dependent mechanisms important for dysphoria from the G-beta gamma-dependent mechanisms important for analgesia, and it rekindled interest in the idea of being able to make a kappa agonist that was directed for analgesia without having the adverse properties. So,
Can I ask you
of
about
course.
the kappa agonists that have been previously explored? Did any of them have the sort of hallucinogenic-like effects of salving?
Yes, yes, they do. We haven't studied it very much. The field hasn't studied it very much. What we do know is that when you look at the clinical reports, the descriptions of what the patients are describing, at high doses, some of them are reporting psychotomimetic experiences, dysphoric experiences. And I think that the kappa agonists that are used generally, like pentazocine, aren't famous for that property. Patients take it for the medication as medication for pain, and they don't talk about hallucinogenic experience, but pentazocine is much less efficacious. I don't know what makes salvia such a strongly hallucinogenic molecule, but I bet it might be a pharmacokinetic issue. Like cocaine being different than methylphenidate, if you can get a rush of drug into the brain the way salvia does, it's different than and the way cocaine does. You get a very different emotional experience than if you take methylphenidate, which does the same thing but at a different kinetics. So we think that dynorphin seems to be important in stress-induced anxiety, stress-induced depression-like behaviors, potentiation of addictive drug seeking and reinstatement of extinguished drug seeking, and all of these effects are blocked by kappa agonists. Wouldn't it be great to see if we could make a kappa antagonist that was a stress resilience medication? What I'm going to describe for you is the struggle with that process. The first being that norBNI is a very long-acting drug. Why is it such a long-acting drug? When we first talked about our results, the medications development leaders at NIDA said, this is great. Frank Fauci said, this is great, but FDA is never going to let us put norBNI into people if you can't explain why it's a long-acting drug. Mentioned this to a community of us at the INRC meeting, Narcotic Research Conference, and we all went back to our lab and tried to come up with an explanation. I'm going to describe the data. I hope I can convince you that we figured it out. What I think is that kappa antagonists have long durations of action because they inactivate the receptor through a c-Jun kinase mediated mechanism. It's a new mechanism, but I'm going to lay out the data to try to explain to you. And the second insight that we had is that it's not unique to the kappa receptor. Morphine, which doesn't activate GRK or rest-independent desensitization mechanisms very well, still produces acute analgesic tolerance, and it does so by a C-Jun kinase mechanism as well. So morphine produces analgesic tolerance by a GRK arrestin independent mechanism. And that the third point that I'm going to talk to you about is that we can separate with this whole strategy of functional selectivity, ligand-directed signaling that's gotten a lot of interest and press in pharmacology literature. If we can identify compounds that are GRK beta arrestin independent, they could be analgesic without dysphoric. And so I'm going to tell you that story. Well, these are the structures of the long-acting kappa antagonists that were developed about 25 years ago. Norbi was the first. Structure is here. It's actually an interesting molecule. It's Phil Portuguese took naltrexone and another naltrexone and put them together. Why he did that, I'm not quite sure. What he says is that he was interested in the idea that two receptors could actually sit together, and by having Norbi, they would, the naltrexone, they could block both of them in a way to look at receptor dimers. But naltrexone, as you know, is a short-acting antagonist, and so the idea that two naltrexones would be irreversible was a surprise. JD-TIC is a totally different structure. It's a molecule made by Ivy Carroll, and it is similarly long-acting. There's nothing about these molecules that would suggest that they would get into the receptor and bind with extraordinarily high affinity, or that they would be cleared extraordinarily slowly, and so it was really a mystery. They do not form a covalent bond with the receptor, and if you put them onto receptors in vitro, it washes off relatively quickly. So why they're long-acting in vivo was not at all clear. And when I asked the medicinal chemists after Frank Voci challenged us, they said, well, it's likely to be a pharmacokinetic mechanism. You know, in vivo, the drug gets into the brain and somehow dissolves in the lipids and somehow stays there for a very long time and is very slowly cleared. I said, okay, any evidence for that? Can you measure it in blood? The answer is no, certainly not for as long as it would take. So it's not detectable, but they said, well, that doesn't mean that it's not there. We just can't detect it. It may be there in a magically low concentration in some spot. There's no way to argue with magic. I mean, we just can't. The second explanation that I thought of, if that wasn't true, is that the difference between in vivo and in vitro is that when you give the drug in vivo, it's metabolized either by an enzyme in liver, enzyme in the brain, into something that does form an alkylation or does inactivate the receptor by a metabolic product that covalently binds to core. And then the third mechanism is that it's not an antagonist in a way that we think of typical antagonists, meaning naloxone binds to the mu receptor and sterically competes for the binding site so that an agonist can't get there, and that's how most antagonists work. That would be different than this one. What if it bound to the receptor, induced a conformational change that inactivated the receptor somehow? That was the third possibility. How do we tell the difference between these three? We did it with a selective protection idea. So if you inject a reversible antagonist before you inject Norbi, the binding sites should all be occupied by the reversible antagonist. The Norbi can't get there, but it'll dissolve and be cleared at the same rate. It could be metabolized and inactivate the receptor at the same rate. But now when you wash away the reversible ligand, you'd ask, is the Norbi still long-acting? And the answer was no. Instead, what happens is that if we put on Norbi to cells in culture, we could look and see Jun kinase activation. And this was a good example of why you should never turn away data that surprise you. I had an undergraduate who was there for the summer. We'd been studying kappa receptor U50 activation of P38, U50 activation of ERK, and now he said, well, we can get some reagents to look at Jun. Let's look at phospho-junk activation. And sure enough, U50 activates phospho-junk, but the Norbi control didn't work. Norbi also activated phospho-junk in the HEC cells. I said, go do it again. Went back, he did it again. Did it again. Okay, undergraduate working in a lab in the summer, how do you rely on that? I said, okay, well, here, but why don't you do it blinded? I'm not going to tell you which is saline, which, he did it, came back the same result. They said, okay, show me transfected and untransfected cells, and then I became convinced. The Norbi activates phospho-junk in hex cells expressing kappa receptors, but not in cells that don't express kappa receptors. And here's an example of how a junk inhibitor can make norBNI into a shorter acting drug. So this is a U50 response for measuring tail flick in a mouse. When you inject the kappa agonist, you get analgesia, which is an increase in the latency to withdraw the tail from a hot water immersion. And then you inject then norBNI at the conclusion of that experiment. And if you do that and measure 7 days later, there's a reduction in the response. But if you pre-treat the animal with the junk inhibitor prior to injection of norBNI, you get a normal response 7 days later. Okay, drugs are great, but junk inhibitors, who knows how selective they are. Let's look at a junk knockout animal. In a wild, U50 will produce an analgesic response in a wild type animal. It'll produce a similar analgesic response in a junk 1 knockout animal. But if we now pre-treat with norBNI, a wild type animal a week later will show a reduction in analgesic response to U50. But in a junk 1 knockout animal, norBNI is short acting. Pretty convincing. But in a junk 2 knockout animal, norBNI is long acting just the same. So the different isoforms of Jun kinase says that norBNI produces its long-lasting effects, and long-lasting effects can be blocked by junk inhibition and can be missing in a junk 1 knockout animal, and it's selective because junk 2 knockouts don't show that same block. Okay, now analgesic response and Jun kinase is fairly ubiquitous, and so could the junk inhibition be influencing tail flick analgesia at some place downstream? This is GTP gamma S assay, and what that is when you activate kappa receptors in the spinal cord, take out the spinal cord, look at the spinal cord membranes, treat the spinal cord membranes with the kappa agonist in vitro, and then you can look at the ability of the kappa receptor to activate GTP gamma S binding to the alpha subunit. It's a great way to look at the ability of a G protein coupled receptor to stimulate GI-GO signaling. And what you can see is that in saline pretreated animals, U69 in vitro will cause an activation of GTP gamma S binding. The junk inhibitor does not block that activation. If you pretreated the animal a week before with norBNI, there's a reduction. But if you pretreat the animal with the junk inhibitor prior to norBNI, the GTP gamma S stimulation by U50 is normal. And that the animal, that the membranes from the animal, clearly, it's at the level of the receptors, what that experiment shows. Okay. Here's a phosphoblot. If you look at the junk 1 knockout animal, you can see that the norBNI will increase phospho-junk in the junk 1 littermate, but it will not activate junk phosphorylation in a junk 1 knockout animal. It's junk 1 selective in spinal cord as well. And so what we're thinking is that ligand-directed signaling of the kappa opiate receptor involves the ability of norBNI to act as a collateral agonist, meaning that even though it inhibits dynorphin and U50 kappa activation of analgesic signaling, it still activates a Jun kinase-dependent mechanism which will block the receptor. And it blocks the receptor for a very long time, essentially irreversibly. This is really interesting. I don't show the slide, but if you inject an animal with an alkylating agent, beta-chlornaltrexamine, which is a naltrexone nitrogen mustard molecule that binds to the kappa receptor, inactivates the kappa receptor, and asks, how long does it take for the kappa responses to return? It's the same length of time. So we think about receptors turning over really quickly, and that's true in transfected cells, and that's true in lots of neural systems where membrane proteins turn over with half-lives on the order of a few hours or less. But kappa receptors turn over really slowly, and if you inactivate all of them in the body with beta-chlornaltrexamine, it takes 2 to 3 weeks before the responses are back to normal. Kappa receptors, for whatever reason, are synthesized at a much slower kinetics. And so that lets us see that kappa receptor inactivation by norBNI is long-acting because the receptors remain inactivated. When we measure with scattered plots the membranes and isolate the receptors, the binding sites are there. There's no loss of receptors. The receptors are not degraded. The binding protein, the Bmax, stays the same. The binding affinity of tritiated U69 remains the same, but that there is no GTP-GAMS activation. Receptors are inactivated. And so I'm going to go back a slide. Well, no, I'm going to go this slide. And what I'm going to suggest is that there is a hypothetical Jun kinase substrate that's an arrestin-like molecule that binds to the receptor after Jun kinase activation and prevents that receptor from becoming activated, and that persists for the life of the receptor. We're in the process of trying to identify and purify that junk-dependent arrestin-like molecule, and have not anything to tell you about further for that. But Kappa receptors are great because they are very slowly turning over, and that makes a lot of sense that we'd be able to see it. But if you were to look from the nucleus up at the membrane, at the face of the Kappa receptor and the cytoplasmic domains that you could see, it would look very, very much like the mu receptor and the delta receptor. They look very similar, a lot of homology. And so we've been struggling with this issue of how does morphine produce acute tolerance when it fails to activate arrestin-dependent mechanisms, as Chris and Mark have shown. And so the question was, could this be also important in mu receptor inactivation? And this experiment shows that in fact it does. Again, we're looking at mouse tail flick analgesia. We're injecting the strong mu agonist fentanyl. We get a robust analgesic response. We've deliberately chosen a dose that gives a sub-maximal response because you could get, 30 seconds, you could get complete analgesia, complete inact. But we chose this dose, and that we waited until the effect wore off, and then gave a second injection of fentanyl, and there was a reduced response. That's acute analgesic tolerance to fentanyl. And as we had shown earlier, if you look at the same experiment in a GRK3 knockout animal, GRK3 knockout animals respond to fentanyl just the same as wild-type littermates, but that the second injection of fentanyl gives you a undiminished response. Acute analgesic tolerance to fentanyl is a GRK3 arrestin-dependent mechanism. But morphine, and here's the frustration, morphine produces a great analgesic response in the mouse, and that when its effects wear out, a second injection gives you diminished response. Morphine produces tolerance. There's no doubt that even though it doesn't produce internalization, it produces tolerance. But that if we inactivate the Jun kinase with the drug, we can block the ability of morphine to produce acute analgesic tolerance. Those same inhibitors do not affect fentanyl-induced acute analgesic tolerance. The regulation of the mu receptor by fentanyl requires GRK and arrestin. The regulation by morphine requires Jun kinase and some hypothetical junk substrate. It's sensitive not to the junk-1 knockout that Norby is sensitive to. In a junk-1 knockout, acute analgesic tolerance to morphine is the same, but a junk-2 knockout animal shows block of the morphine acute analgesic tolerance. And both groups of animals respond to fentanyl just the same. I don't know if I have this slide. I may have taken it out. Let me go back and describe one more experiment to you. If you treat the animal with morphine and then treat the animal with fentanyl, you'll get cross-tolerance, full cross-tolerance. If you treat them with morphine, the cross-tolerance would be blocked in a junk-2 knockout, but not in a GRK3 knockout. And if you did this experiment with fentanyl first, you'd get cross-tolerance to morphine, and the morphine tolerance would be blocked by GRK3, but not by junk knockout. So if you followed what I just said, when fentanyl inactivates the mu receptor, it does it in a GRK3 and arrestin-dependent manner. When morphine activates the mu receptor, it inactivates the mu receptor in a arrestin-independent junk-2 dependent mechanism. And that's really interesting because it's never been described before. I believe it. Go ahead.
Would you speculate that that might be something that pain docs often talk about, opiate rotation as a means of dealing with long-term tolerance?
I don't know, because if you've treated with one and you produce tolerance, you'd be tolerant. If you treated with fentanyl first, you'd be tolerant to morphine also. I think the rotation depends on the selectivity of the drug, and so it may have to do with kappa mu differences, Or if you were in Gavril Pasternak's lab, you might talk about mu1, mu2 differences, but I don't know what rotation is really doing in this model. You may have a...
I've always speculated that they have slightly different lipophilicitys and they go to slightly different receptors because we can inhibit pain in so many places, cortex, spinal cord.
So we're highly motivated to try to understand more about the June kinase pathway for two reasons. One is we think it's likely to be general. Ken Mackey tells me that a very similar relationship exists at the cannabinoid CB1 receptor between THC-induced tolerance and Win-induced tolerance, Win being GRK dependent, THC being a junk-dependent mechanism, and he's working to try to define that. I believe it could be general. For things that are inefficient at activating G protein receptor kinase, it could be an alternative mechanism. I don't know what it means physiologically because I don't know what the endogenous dynorphins do yet, whether they activate a junk-dependent mechanism or not. We have to still study that. But I'm also excited about this for a second reason, which is that when arrestin gets recruited by fentanyl or by U50, there's, I told you, arrestin-dependent signaling events that compounds that don't activate arrestin will not activate, not do, but there may be junk-dependent signaling events that we have not yet defined, and I'm interested in trying to understand those. So that's what I'm proposing here, possible junk-dependent signaling mechanisms that we just don't understand. So we're going to see what happens with that. But what we now know, this study, this simply says that it's not unique to fentanyl and morphine. When you look at methadone and oxycodone and other strong agonists of this sort, they are arrestin-dependent inactivation. But morphine, buprenorphine, M6G are able to activate the June kinase mechanism. And this could be a relationship to efficacy, but exactly whether it's efficacy or not, we don't fully understand. It may have something to do with efficacy because these are less efficacious compounds than these. But whether efficacy means what we used to think it means is still something that people in the opiate field are trying to tease apart. But the possibility of two different forms of biased agonism exist. Okay, so let's go back to looking for a new Kappa antagonist because of all the therapeutic potential. The Kappa antagonists, when you give them like JD-TIC and they have their long-lasting effects, that might be really great because if you've got somebody who you would like to maintain as stress resilient, 3 times a day dosing is not ideal, whereas once a week dosing might be really ideal because they don't have to think about, oh gosh, I'm going to be stressed today, I better take my pill. They don't have to think about it at all. And so long duration of action is not bad as long as the activation of June kinase and the pharmacokinetics can be understood. The problem is if an individual takes a long-acting medication and they develop problems, it takes a long time for them, for those drug effects to wash away. And for that reason, drug companies would really rather deal with a short-acting compound. And so we got every Kappa antagonist we could get, which comes from all the ones developed by Ivy Carroll at the Research Triangle Institute, all of the ones developed by Eli Lilly and McLean, and some compounds that are not selective, but they are Kappa antagonists as well. And we asked, is there a relationship with the ability of these compounds to produce antagonism and June kinase activation, the duration of effect related? And the answer is yeah. So if you look at the duration of their antagonist effect after injecting a mouse and then challenging with U50 to look at analgesia, how long does the effect last? It exactly correlates with the amount of June kinase activation in the spinal cord of those mice. And so we now have an ability to distinguish those short-acting compounds from the long-acting compounds and do drug development of this and ask, are these better tolerated by humans than the long-acting compounds? Yeah.
Wondering the June kinase phosphorylation, can you see it for a very long time or just very short period of time?
It's a very short period of time. So if you look at the spinal cord and then harvest the spinal cord, the effect is evident within 24 hours. It's completely resolved within 48 hours. And then if you come with a second injection of NorBNI, there's no increase in phospho-junk. So repeated injections of phospho-junk fail to activate June kinase more than once, probably because the receptors are inactivated. and so a subsequent treatment won't. Yeah, that's your question. There was another question.
So morphine activates that same pathway, but
Yes.
its tolerance doesn't last for 3 weeks. So
And if you were to inject an animal with an inactivating agent, alkylating agent, beta-chloronaltrexamine will also inactivate the mu receptors, and come back and ask, when does the morphine response comes back? It's fully back in 3 days. The mu receptors in vivo turn over much more rapidly, and the analgesic receptors required for morphine analgesia turn over more quickly.
Joe, do you see the same thing in cells as opposed to in vivo? You've got all the issues of blood-brain barrier, tissue penetration. Do you see it in HEC cells, for example?
We do see it in HEC cells, but the HEC cells are rapidly dividing. I think they've got a 48-hour turnover rate. So if you look at it, the actual duration of inhibition is more on the order of 24 hours that the Norby lasts, and that if you were to inhibit Jun kinase, you can shorten that significantly. So we can see the inactivation in HEC cells, and that's where we're trying to purify the junk substrate, but it's not the 3 weeks because cells divide more rapidly. When we look at NG108 cells or PC12 cells, which divide more slowly, it's similarly longer action. It has to do with how quickly those cells are synthesizing receptor, and they do so because they're transfected at a much higher rate than neurons do. Other questions? Yeah.
Maybe you mentioned this, it's sort of related to that question. So you show that the antagonist has a long-lasting effect where the receptor is just inactivated, nothing will activate it. How is the activation restored? Is it part of the signaling pathway or is it receptor turnover? Like
No.
a week out, you start seeing a response to activation of the Kappa
Um,
opioid receptor.
Yes. So the answer is when we challenge with the agonist weekly, so at 1 week later there's a partial return, there's almost no return. 2 weeks later it's partially returned. 3 weeks later it's nearly completely returned but still significantly down. 4 weeks later it's fully back. So we can measure kinetics of recovery of the U50-induced analgesia that matches what we think is the chloronaltrexamine, the synthesis of receptor. If we were harvesting tissue at those 4 time points, the receptor number, the Bmax, would be unchanged.
So it's receptor turnover
It is receptor turnover. As if once the receptor gets junked, it's inactivated irreversibly, even though, well, we don't know why. Okay, so we can divide ligand-directed signaling. Again, this is a...
Charlie, one quick question, maybe I just missed a bit. We're talking about an antagonist with this long-acting, but does it activate acutely?
Acutely, it acts as a competitive inhibitor. So if you, in cells in culture, if you put them together, it works just like naloxone sterically. It works at the same binding site as the agonist, but fails to activate G protein signaling. So acutely, it's sterically inhibiting agonist binding.
Jun kinase.
But it's activating gene kinase at the same time through a different mechanism, in a G protein independent mechanism. And we're teasing apart the mechanism.
Does it promote kappa receptor endocytosis?
It does not. No, it prevents endocytosis completely.
It just doesn't activate GTP gamma.
Does not activate GTP-MS, doesn't activate G proteins at all, doesn't produce analgesia. It's not a partial agonist, if that's what you're thinking. It's not a partial agonist.
No, what I was thinking is if it's actually functioning as a bivalent ligand, as Gazy was initially
Right.
proposing, you might expect, because many clustering compounds will affect endocyte
Right.
trafficking, and
So
it might trap the protein in a different membrane.
So when Phil presented that speculation that it could be acting as a bifunctional ligand, people did the measurements and said, look, it's not big enough. And so he went back to the lab and made bigger spacers, and then he has bifunctional ligands. But Norby is not a bifunctional ligand. It's not any bigger than dynorphin, which is has a C-terminal tail, and it interacts with
So knowing
other
the
dimensions.
diameter of the hydrophobic bundles, it could never.
And also JD-TIC.
Really small. And when we look at the crystal structure and how JD-TIC fits into the receptor, it's not spilling out into the extracellular domain. It is likely to be inducing a conformation that activates Jun kinase through alternative non-G protein dependent mechanisms, and we're teasing that apart. What are the upstream to gene kinase, and then what are the downstream effects of the signaling? Okay, so ligand-directed signaling is really remotivating the pharmaceutical industry in many, many dimensions. The idea that you could have a functional selectivity where you get a set of agonist effects with one compound, but the ligand binding to the receptor initiates a different signaling with a different, forces us to re-examine compounds that we have. And that's kind of summarized in this cartoon. I imagined that high efficacy compounds would be able to activate the p38 dependent aversion mechanism, but partial agonists that don't do this would be analgesic without being aversive. And that you could separate long-acting kappa antagonists from short-acting kappa antagonists based on gene kinase activation. And we're seeing evidence that this is likely to be true, and we're seeing evidence that there may be new classes of compounds available.
Charlie,
Yeah.
maybe this is off target a little bit, but I wonder is there any connection here with inverse agonism?
Well, inverse agonism is an important concept. It's typically dependent upon constitutive activity of the receptor. So you have typically constitutive activity when you have overexpression systems and receptors are not. There's not a lot of evidence, please disagree if you believe otherwise, for constitutive activity of the mu and kappa receptors when it's expressed at normal endogenous tone. You can see naloxone given in vivo to having inverse agonist activity. I believe that that's because of basal tone of endogenous opioid peptide, as opposed to the receptor being constitutively active. Is that consistent with your idea?
It is, because if you knock out pro and kappa, you don't get naloxone aversion.
So there are other systems, and when you talk to Brian Kabilka, he talks about having constitutive activity in some of the beta-adrenergic systems that he studies. So constitutive activity is not heretical. It exists for other systems, but not in the opioid system. So I didn't list it in here because I don't have an example of an inverse agonist. Requires constitutive activity.
But it's an interesting question that goes more to conformational dynamics. Like Brian's work has shown very nicely with the beta receptor that the receptor can populate many different conformational states, and that an inverse agonist sort of restricts the conformational space to some degree. And then the idea would be, and this goes from the work from Michel Bouvier, for example, that some of those conformational states may have alternate signaling properties.
Yes.
So, and this is maybe an explanation for why a compound that's been considered an inverse agonist based upon, say, a G protein mediated readout could actually be what I think Charlie would call a collateral agonist, in the sense that it's actually able to stimulate another pathway.
So Mark and I have talked about efficacy in lots of different contexts, but what our original concept of efficacy was very linear. The ability of a ligand to efficiently activate G-beta gamma production would be opiate efficacy. But now when we've got multiple pathways, the concept of efficacy has to have multiple dimensions as well. So you have pathway selective efficacies depending upon which output you are looking for. So there's a lot of range there as well. You could imagine it be very efficient in one pathway, less efficient in another. Norbion is an example, very efficient in one pathway, not efficient in the other. Okay, so Ray Stevens and his colleagues did crystallize the kappa receptor with complex with JD-TIC and speculate how Norbion I fits into the crystal structure. There's a lot of information now about the way the ligands bind, and so the question is, what's different about the different forms of signaling, and can we make sense out of the pattern of binding pocket folding around the ligand that will then tell us how a compound is either a pathway selective for one or another Okay. We went back because of the hypothesis that you could imagine having a compound that would be beta arrestin independent, and went back and looked at every available kappa partial agonist that we could find, including, and compared it to U50, which is a full agonist. And we come up with a way to measure kind of the pathway selectivity or bias based on the ability of the receptor to be activated in a G beta gamma way, which is a p-ERK activation, or a p38 way, which is an arrest independent, and the ratio of arrest independent to arrest independent varies strikingly among the different compounds that we can look at. None of these are ideal because none of them are really selective for the kappa receptor. So if you give them in vivo, pentazocine will also activate the mu receptor, buprenorphine, butorphanol will also activate mu in addition to kappa. But we just wanted to get a sense of, is there any proof of concept that you can get a separation? And the answer is they range pretty widely. And the best example is nalfurafine, which is a compound that was developed by Toray Pharmaceuticals in Japan. They use it in vivo as an anti-itch medication for individuals with renal failure on dialysis. The buildup of the toxic metabolites in their blood creates a huge irritation in their skin. And what you can see is the ability to activate G beta gamma is 200-fold more potent than the ability to activate P38. And so the idea that you can get selective compounds, pathway selective compounds, if not receptor selective compounds, at least is demonstrated there. And the idea that we could actually make pathway selective and receptor selective compounds means that there may be a new class of analgesics. The advantage of a kappa analgesic that's not dysphoric is that it would likely fail to have the addictive properties of a mu agonist. This simply sketches out the way a pharmacologist thinks about pathway selective efficacies, and I won't go into that with you because it's way beyond an addiction course, but here's a signaling cascade that summarizes what I said already, having to do with the ability of different pathways to mediate analgesia, anxiety, dysphoria, long-lasting antagonism, which in a cartoon mode talks about ligand-directed signaling in a way that makes graphical sense. Summarize what I already said, and then thank the people who did the work. Okay, so thank you so much. Any more questions? Good.
I think that's super cool. You guys are getting close to what could actually be a therapeutic potential with this complexity of agonist effect.
What I love is the idea that we can get some of these antagonists into clinical trials and really see how they make people feel. The prediction from the animal studies are really strong, but until we get them into people and ask, does it affect stress resilience? Does it affect cognition? Does it influence the way you feel in a way that is predicted by the animal study? We won't know whether animal models are accurate or inaccurate. And if we get into cognition, as we were talking about earlier, it's very difficult to measure psychotomimetic effects in animal models, but they're easy in people. They'll tell you.
So they could work for anxiety and depression as well.
You know, you might remember this, but Lars Terenius in the mid-'80s, late '80s, was very interested in the role of dynorphin in cognition, and he was doing what was popular at the time, which is sampling CSF levels of everybody he could get to sit still. And he measured dynorphin levels in schizophrenics, and he found that dynorphin levels in a class of schizophrenics were really elevated. And those were individuals who had auditory hallucinations. And he gave those individuals, in a kind of a double-blind crossover design, high doses of naloxone, and found that the auditory hallucinations of individuals with high doses of high levels of dynorphin were really affected by naloxone. It'd be really interesting. They did a larger trial, and it wasn't generalizable to all schizophrenics, of course, but it would be very interesting to go back and look and see if there could be a role of dynorphin in cognitive disturbances that might be attractable with a good kappa antagonist. I'd be intrigued by that.
I heard, I might be totally wrong, but I think I heard this from Mary Jean Cree, that naloxone and maybe even naltrexone are actually weak or maybe partial kappa agonists in addition to antagonizing mu receptors. So they bind, they occlude dynorphin or other agonists, but they actually activate just.
So Mary Jean is quoting Jean Bidlack, who did a cost cell expression study at high levels of receptor and looking at GTP gamma as activity. So when you've got high levels of receptor, you're much more likely to be able to see weak partial agonist effect. But in none of the electrophysiologic experiments that we've ever done, when we wash on naloxone, do we see any evidence at physiologic levels of receptor expression for a partial agonist effect of naloxone. So I think she's enthusiastic about the in vitro studies, but I'm not sure that it's physiologically relevant.
When you think about it, from what we now know about what we're learning, thanks to some of the beautiful structural work that Charlie alluded to, of how these receptors actually operate, it's almost a miracle that any compound could truly be an antagonist. Because to be an antagonist means you do absolutely nothing to the receptor, except you bind to it and you prevent any other ligand from binding to it. That's
Really high.
and do absolutely nothing to the conformational landscape that the
Which is
receptor
almost-
lives in.
Yeah, so you expect that maybe there's some phosphorylation of that you're getting with norBNI, because you just changed the
Yes.
conformation and now you've got a site which you can phosphorylate and change.
It's not fully an accidental. I understand your point exactly, but the medicinal chemist first got a clue that there were opiate receptors that were there. I mean, the idea that there was an opiate receptor was based on looking at the structure activity of a range of compounds, including nalorphine, which is a very, very weak mu partial agonist. And they said, if it can block morphine effect, very low efficacy, it's likely that there's a binding site for it. And based on that information, they went and refined with more synthesis until they searched for and found one that was truly neutral, like naloxone, naltrexone, and nalmethine. So they knew what they were looking for, but the clue was nalorphine, which was an extremely weak partial agonist.
So it's an interesting question then, how to relate this, the pharmacology that's emerging from, and all the selectivity that's emerging to in vivo physiological significance of this. And I think that's an open question. I mean, there are some receptors where constitutive activity is really physiological, like melanocortin receptors.
I
Where
don't.
they have very high constitutive activity, and they have a natural ligand that's actually an inverse agonist that reduces activity. But for many, we really don't know the function of constitutive activity, or if it's a relevant parameter.
So you notice there's a whole two other levels of complexity that I didn't even touch on, which would be splice variants of the receptor, which have been hypothesized, and possible receptor dimers that could influence signaling properties. And I haven't begun to think about how those would affect what we do. I mean, it could be that splice variants or alternative forms of the kappa receptor could alter signaling properties, or binding partners, dimeric partners or multimeric partners could influence signaling properties. We haven't even touched that. I don't know.
So Charlie, there's some data when you block the kappa receptor that, for instance, you don't get the aversive effects of marijuana. So you wouldn't get, so as a drug, as a drug therapy, let's think about it as a drug therapy, you may have problems with people not having this opponent process which may limit their taking of the drugs. I know it prevents the stress-induced relapse of cocaine, but do you think it would actually have people taking more drugs, or do you think they would take less drugs because they wouldn't have the stress-induced relapse?
Well, it depends on whether you believe Koob. So you might want to comment about that, because alcohol would be something that you'd be less likely to take if you had a norBNI on board. That's what Brandon Walker talked about. But if you are motivated to stop taking the drug because of the dysphoria that you're fearing, then you're more likely to take more drug. That's Chris's point. And Jennifer Whistler, I think, showed some data with ethanol that norBNI might increase ethanol consumption. We haven't seen any of that in our rat self-administration data where animals with norBNI take more cocaine. It doesn't affect their cocaine consumption.
But if you look at place take away the Kappa receptor, you only have the rewarding effects, you don't have that aversive component, which...
And you could imagine.
I don't know. What do you think, George?
Well, I mean,
I think it's an interesting
I
question.
think it's always going to be an issue. I mean, if you give diazepam to alcoholics,
Yeah.
or to people taking alcohol, they'll drink more alcohol. That's been known since Tony Schillich published 40 years ago that diazepam will facilitate drinking of alcohol, but diazepam also alleviates alcohol withdrawal. So I think it's going to be one of those situations where we're going to have to see how these play out clinically. I mean, as Barbara didn't mention it, but there is some data that gabapentin can actually in animals facilitate social drinking in animals, but she didn't see any abuse of gabapentin in the clinical trial. It actually helped the patients and blocked the excessive drinking. I'm personally not that convinced that the punishing effects of drugs have that much of a beneficial effect in limiting drug intake. I don't know, Paul Kenny may want to speak about that because there's punishing effects and punishing effects. I mean, in the habenula, that might be a different story, but I'm not convinced that, maybe some people don't like opiates because they make them throw up, but an opiate addict says it's the best throwing up they ever did. So I think, you have to be...
Yeah, I think it's going to be
Keep an open
interesting
mind.
to see what happens on that.
I don't know, well, what do you think? I mean, do the aversive effects of drugs limit their intake?
It's kind of mixed. For smoking, you have to develop tolerance for the aversive effects
Yeah,
before you really smoke. Well, I'd argue though, I think my thinking that's been it's very simplistic. I think if you give an animal a high dose of nicotine, it doesn't like it, for example, or cocaine even a high dose, and it's punishing and aversive. But I think where aversion comes in is actually in the concept of satiety. How does an animal dictate how much nicotine it's going to take, or how does a human drug user say, I'm going to use this amount of heroin today, that's the amount I need. And I think that's where aversion satiety comes in. Aversion is actually the extreme form, I think, when you pass satiety. So one way to control drug intake is make their satiety levels, they reach those quicker. So I think the systems that are involved in aversion are probably the endogenous satiety systems. So that may work, but I think global, give them antabuse or something, is not going to work. They're addicted to the drug, they need it. But the systems the drug is working on, that may be a clever way of doing it. So it's something we're working on right now, for sure. What's the relationship between satiety and aversion?
So we've done a nicotine CPP dose response curve, and it's like you would predict a U.
Yeah.
You've probably seen similar things as well. And the Norvion will increase the preference at the high doses. So you're exactly right.
Same with marijuana.
It's exactly right. But yet, how that's going to play out for people.
Yeah. Well, we have the human literature. So we have, like we heard last night, with the alpha-5. And that very specifically works just if you have a measure of aversion, reward-based aversion. So not global kind of malaise, but the animals demonstrating anhedonic-like states. That's what alpha-5 does. As we heard, it feeds in probably from the IPN indirectly into the midbrain dopamine systems and shuts them down. And so that has huge effects in human behavior. It dictates whether people are going to become smokers to some degree and how much they're going to smoke. So we know that those systems play a role in humans. It's how we manipulate them pharmacologically for therapy.
So you're right, it depends on where we get our funding from. We get it from United, but it stops addiction. If we get it from the drug pushers, if you get some funding from
Columbia.
you can get some Columbia money,
The way the rate is going, that may be better funding.
it may be a bit more stable. More stable funding. Okay, so a break?
Yes, that sounds good. Yeah, and we'll start at, well, actually, yeah, let's just have a 5-minute break.
Thanks.